So next up, we have Dr. Andrew Dhawan. Andrew, known for several years, and he's a neurologist, and what he specializes in is genetic disorders, not only neurofibromatosis, but some other genetic disorders. And he also is in the lab doing research. So he is going to talk about tweaking the gene editing in genetic disorders. So thank you very much, Dr. Dhawan.
Thank you so much, Dr. Sheesh, and thank you everyone for the opportunity to speak here. It's truly an honor and a privilege, and I feel so lucky to have this opportunity. Um, as was mentioned, I'm a neuro-oncologist and a physician scientist here at the Cleveland Clinic. My work focuses on inherited brain tumors and inherited syndromes affecting neurologic health. So today I'm going to speak to you about a few stories that all center around this novel concept of tweaking the gene or gene editing in genetic disorders. I have no relevant disclosures to this talk. I will speak about some medications, but I have no disclosures relevant to that. So first, we'll take a step back and we'll talk about what is a gene. We'll talk about how gene editing works, how it could be implemented in humans, whether this could actually be a cure, and I'll give you some places to find more information about this. So genes are segments of DNA, and if you look up the definition, you'll find that it says the unit of heredity. And what this means is, to take a step back, is our body is composed of trillions of cells. If you zoom in on each of these cells, at the center, you'll find something called the nucleus. Within the nucleus, you'll find these X-shaped objects, which are called chromosomes. And these chromosomes are actually wound up molecules called DNA. And when we identified this, DNA, we found out, was comprised of 4 bases, A's, T's, C, and G's. They're 4 letters. And these form a sequence, 3.3 billion base pairs long. And what was found is that genes are really these lengths of DNA, these short segments, okay, that are the instruction sets to make RNA and proteins. And proteins, of course, are the end effectors of how the cell accomplishes its ultimate functions, how it does what it's supposed to do. So these are the recipe books or the instruction sets, the instruction manuals for our bodies. It's all contained within the DNA. And so what was later identified was that changes to these instructions, so spelling mistakes or losses of certain pieces of this DNA, are actually what the cause is in families that have disorders that span generations, like NF. And previously, we used to think that DNA was unchangeable. What you were born with was your destiny. But that's not true whatsoever, and our much research has focused around showing that. So what you may have heard about in terms of DNA and gene editing in humans is this concept called CRISPR, Clustered Interspersed Short Palindromic Repeats, which is a long acronym, which really just means that it's a find and replace for the genome. So like I mentioned, the genome or the DNA of all of our body is contained in chromosomes, and those form the instruction sets. Well, this is how we change those instruction sets. We look for a very specific sequence, and then we can cut it out and replace it with another sequence. And that's what CRISPR enabled. So CRISPR won the Nobel Prize in 2020 because it's such a transformational technology. And it actually comes to us from nature. It's a defense system that bacteria use against viruses. And so we have been able to take that system and implement it in other cells. And here's how it works. So a cell which contains all of the DNA is transfected with 2 things. One is an enzyme complex, so that's another protein, which contains a guide molecule, okay? And that's going to be the find part of our find and replace. That tells us exactly what are we looking for in the DNA sequence of the cell. It also contains a DNA cutting enzyme and a healthy DNA copy. And the concept is very simple. Basically, the guide molecule takes the enzyme to the exact part of the DNA which has the problem. That's the find part. The cutting enzyme then cuts out this defective strand, and it is replaced with the healthy strand. Pretty simple, right? Well, it turns out it's actually quite complicated because this would have to reach every single one of the trillions of cells in your body in order to have an effect. Um, so the question then comes up is, do all cells need to really receive editing for someone to receive benefit? And it turns out that that's probably not true because only some cells are the problem. For example, in various blood disorders, really it's just the bone marrow or the blood cells that are the problem. And so what we can do is then remove the cells that are the problem, correct those cells, and then re-inject them. So that's one strategy. Other strategies involve targeted implementation of CRISPR, so using direct injections to specific tissues like the heart or the brain. Or people have also been studying how we might be able to target all cells systemically, but that is proven to be a greater challenge. And just to give you a sense of how challenging this actually is to implement CRISPR in a human system, this diagram shows you all the types of strategies that have been tested. And each of these things on the end of this circle are different types of methods by which scientists have been trying to get CRISPR into cells to actually achieve genome editing in humans. This is not an easy task, but as you can see, we've been working very hard to make it a possibility. So as we've discussed, some challenges in gene editing technologies include one, the targeting of all cells or just some cells. And the other thing is when you start to inject foreign substances into the body, which contain DNA, your body reacts to this and it tries to reject it. And so a lot of work has focused on how do we actually suppress the immune reaction or the body's reaction to injecting these foreign substances into the body. And so there are challenges with immune reactions. And it's unclear whether these may actually give you benefit later in life. For instance, we think it may actually confer greater benefit in people who have a lot of life ahead of them, so young children or babies, because they haven't had enough time to develop the challenges associated with the genetic disorder. And the other part is sometimes the find part of that find and replace doesn't work as perfectly as we want it to. Those are called off-target effects. So it might match something with a very similar sequence, but not a perfect match. And we've been working on how to make that exactly and more precise, but that does remain a challenge. And I think that's actually probably one of the greatest challenges of this technology and why we're so careful about bringing this to humans. So despite all of these challenges, I'm going to share with you quite two stories actually that highlight the success of CRISPR therapies, which I think are super exciting. And this story comes to us from December of 23 or 24, where the first CRISPR therapy was approved for use in humans with a drug called Casgevy. And this was for a blood disorder called sickle cell disease. Patients with sickle cell disease unfortunately experience very significant episodes of pain throughout their life. These occur at random, and these patients have to go to the emergency department with crushing chest pain, severe head pain, severe bone pain. That's probably the most severe pain any of us could ever imagine. So preventing this painful crisis is something that was on top of everyone's mind. And because this is a blood disorder, the thought was, well, we can take out the blood cells, replace the DNA with healthy DNA without sickle cell trait or sickle cell disease genes, and allow blood cells to function normally, thereby preventing painful crises. And this study showed great benefit. Of the 31 patients treated with this drug, 29, so 93.5%, had freedom, complete freedom from painful crises for at least a year in the 2 years following treatment, which is pretty much unheard of for any other therapy for this disease. So this was a huge success. So it's now on the market. It was FDA approved for sickle cell disease. And I think this shows a very strong example of how CRISPR therapies are going to reach the market. It's going to be first in targeted things, most likely blood disorders, because we know what cells have to be targeted. And there's a very clear endpoint of reduction in painful episodes. So, as mentioned, this was approved as the first CRISPR therapeutic. The second story is actually from 2 days ago, just to show you the sense of progress in this field. If you read the New York Times, you'll find this article. baby is healed with the world's first personalized gene editing treatment. And so this is a really, really exciting story. And let me tell you the story. So this was a baby where both parents had a gene for a urea cycle disorder called CPS1. Okay. And basically, these kids don't do well in life and they develop very abnormal levels of a toxic chemical that builds up in their body and causes brain dysfunction and multiple organs actually to have failure. There's no treatment for this disease. And so, what researchers did was as the baby was born, they identified the changes in the genes from mom and dad that cause this disease and that were present in the fetus. Okay. And what they did then is they created a mouse that had these exact same changes in its DNA. And so the mouse had the disease, and they could test CRISPR therapies on the mouse to make sure they were safe. And luckily, we can do this in many mice to pick the optimal therapy because I showed you how many ways there are of delivering these treatments. And so they tested batches and batches of these therapies in mice, and they were able to show that the corrective treatment, the best one they could choose, actually worked in the way they wanted it to. They tested it in human cells then, and then they also tested it in non-human primates, and they looked for off-target effects in all of these other genes. So they tried to make sure that that find and replace, that find, was very specific for those changes that came in that baby from mom and dad. And so this is a timeline of these of the progress of this research. So the baby was born, okay? They developed the patient-specific cell line in the second month of life. The mouse model was made at month 3, okay? They proved all the off-target effects in month 4, just to give you a sense of progress. They showed it wasn't toxic in month 5. Month 6 and 7, they were testing it in non-human primates, so macaques usually, like monkeys, and to show it was safe and it worked. The FDA then approved it in month 7 in a rapid emergency use situation, and the patient received his first dose in month 7, did well, but then suffered a respiratory illness, and they noticed that the levels of that toxic chemical were reaccumulating around day 22 after the injection. And they did a second dose and showed that he's been doing just fine after that. And he's essentially what we consider now biochemically cured from CPS1. So he's the first patient in the world to receive a patient-specific gene editing treatment after birth. And so the authors very excitedly wrote in this New England Journal paper that was obviously released earlier this week that we anticipate that rapid deployment of patient-specific gene editing therapies will become routine for many genetic diseases. And I think all of us here know that NF1, NF2, other metabolic disorders without any treatments are those that we are laser focused on. And the fact that this paper provides a roadmap to show how it can be done in a safe and efficacious way in just 8 months with today's technologies is impressive. This work required a huge amount of funding and collaboration. It was done at Harvard, and it was done with the support of the NIH. And so this work, I think, is just an incredible testament to the science that's happening in this field and how quickly this field is changing, and to show you all that there's actual hope. So to conclude, gene editing is a very promising approach for NF, and as was mentioned earlier today, there is the NF Gene Replacement Initiative, which is working on very similar approaches and analogous technologies. As I mentioned, safety signals will be important in early clinical trials for these sorts of therapies. Being treated early in life, I think, will be most important. NF is a challenging disease, though, because it has many possible changes in the DNA, so it will require variant-specific treatments. There isn't going to be an off-the-shelf treatment for NF because of how many different ways NF can be caused. So we're going to be stuck with most likely personalized treatments, and so it's going to take some work to figure out how we can get this to scale to reach every patient in the world in a timely manner. And we don't know yet, obviously, the long-term effects of these treatments. as I mentioned, that study came out just 2 days ago, and that baby is less than a year old. So we're going to find out what happens with time. But this field is progressing at a rapid pace. More research is obviously needed. And so I thank you all for the opportunity to tell you these stories, for the opportunity and the incredible, bravery that you show in the face of this disease. And I think, a lot of this sounds like science fiction, and you might hear things on the news and different things about science, but it's important to be objective. And I love this quote from Marie Curie, one of my favorite scientists who inspires me. Nothing in life is to be feared. It's only to be understood. And so now is the time to understand more so that we can fear less. So always ask questions and go in with a mind of understanding. Thank you, everyone.
Terrific. That was very interesting, kind of an exciting discussion there, Dr. Dhawan. Thank you.